Okay, this video is on carbohydrates. Carbohydrates is a general word. It encompasses monosaccharides and polysaccharides. Pause the video and have a read of all these questions. See what you can answer now, and then when you go on, you'll see, your new learning. So pause the video now. This is glucose. It's the monomer of a lot of polysaccharides. Can you draw a glucose molecule? So if you're asked to draw a structural formula for glucose during the exam, what would you do? Where would you look? So obviously look in the data booklet, and if you looked in the data booklet, this is what you would see. So this is a glucose, an alpha glucose, so we're going to look at what alpha and beta means. Okay, now this alpha glucose is not drawn completely. You need to be able to add the numbers of the carbons, how many carbons there are, and they're missing hydrogens. So each carbon's got 4 bonds, so there's 1, 2, 3. So clearly this has got a missing hydrogen. And work out the formula for glucose. So if you number them, you'll see that there's an oxygen in the ring. This particular CH2OH is a group that we use to work out the positioning of the other OH groups. So we're going to look at on either side of this oxygen, and the carbon, what's going on around carbon 1 and what's going around carbon 4. So at the moment, the reason that this, as well as this is a 6 carbon monomer, there's also 5 carbon monomers. So the formula for glucose is C6H12O6. So the general formula for all monosaccharides, just monosaccharides though, is however many carbons you've got, it's double the hydrogens. So you can see this is double. So if you had a 5 carbon sugar, then the hydrogens there'd be 10 and there'd be 5 oxygens. Once you put them together though, once you start linking monomers to make a polymer, you lose a water molecule every time you do it. So this formula doesn't work for polymers. This formula works for everything. So you could see that if you have 6 carbons, you've got also 6 water molecules. So you can get to this formula using that general formula. Alright, the reason that this is called an alpha glucose is that on the first carbon, the OH group is pointing in the opposite direction to this CH2OH group. So alpha is the beginning of the alphabet, like alpha with our fatty acids. It's the first carbon right next to the carboxylic. This is the first isomer, and they're on the opposite sides. Okay, so if these are all chiral carbons, so there's this carbon here has got 4 different structures on it. So if it's got 4 different bonds on it, then it's a chiral carbon. This one is 2, this one is 2, this one is, and this one is. So there's 5 chiral carbons where what is attached to them can flip in the opposite direction. So they're all stereoisomers. So if this flips around, then the light would behave differently and the molecule behaves differently. And so they're different isomers of each other. The last carbon up here, the 6th carbon, has got 2 hydrogens on it, so it doesn't have 4 different things on it. So a beta glucose is when you flip that OH. So now the OH and that CH2 are both in the same direction. So just when we did cis, we got the smallest ones on the same side, like sisters. A way of remembering it might be B cis, same. B sisters. So the B ones are on the same side as this CHOH. This CH2OH we'll use later on. It's not got an OH on the other side, it's just got a hydrogen. Okay, so that's alpha, that's beta. Alpha, beta, alpha, beta. Okay. You can sometimes, occasionally, very rarely, but you can actually show a glucose as a linear molecule. So if you ever see this, all those OHs, just remember 6 carbons, it's a glucose. another way of drawing glucose, so a structural isomer. So alpha and beta, remember, are stereoisomers of each other because the OH group on the first carbon, the first chiral carbon, and then glucose has 5 chiral carbons. So have a look at galactose. What do you notice is different between the glucose and the galactose? Could you explain that in words? Alright. So glucose and galactose both have the same formula, C6H12O6, but have a different arrangement about the chiral carbon 4. So 1, 2, 3, 4. So glucose has the oxygen down. You'll see this in the data booklet. Galactose, and you should write this in the data booklet, galactose has the OH up. Otherwise, this one, the ones I've picked, the OH here are both O up. So this glucose, can you remember what this one is when they're both on the same side, like sisters, B, cis? That's a B glucose, this one. Okay, so they're stereoisomers when this one is on the different side. So note that in your data booklet. Alright, fructose. Fructose, what do you notice about the ring? So instead of having 5 carbons, it's got 1, 2, 3, 4. So this one is like the other one with glucose. This is the one that we used to compare our OH group to. Okay, so that one was over here on glucose. It was sticking up. And if this OH group was in the same structure, same direction as that one, B, same, sisters, then this was a B. So this is a B fructose. So it's fructose because it's got only a 4 carbon ring. Has the same formula as glucose. So it's a totally different structure. It's a structural isomer. Different structure, but it can also be an isomer. So this was a B fructose. So clearly this entire thing, if it flipped, that would be an A. So how many chiral carbons has this one got? So if we have a look, this one's got a hydrogen here. So it's got 4 different groups. So 1, 2, 3, 4. These ones aren't because they've got a carbon with 2 hydrogens. So those 2 are the same, and those 2 are the same. So they're not chiral carbons. So it's only got 4. And alpha, can you work out? I've talked about it. Can you remember? Alpha is when this OH is on the opposite side to that. Opposite side to that. Okay, I'm probably going into too much depth about beta and alpha, but you need to sort of recognize that these groups can flick around and then it makes a different stereoisomer. Okay, disaccharides. Di means 2, so it's when 2 monomers, 2 monosaccharides get together to make a disaccharide. Okay. What are the names? Which particular monomers are these? Okay, so which particular monomers? If you have a look at this, this is a 5 carbon ring. So clearly it's either glucose or galactose. So if it's glucose or galactose, you look at the 4 carbon here. Which way is the OH pointing? So if you look in your data book, you see that it's coming down here. So this must be a glucose. So to work out if it's alpha or beta, the oxygen's here. This is your CH2OH with just a hydrogen on the other end. This is the marker. So then you have a look, is your OH down or up? So it's down, so it's in the opposite direction, so it's alpha. So this was alpha glucose. This one, you can look in your data booklet, this is fructose, because it's a 4 carbon ring. So your CH2OH, like this one, with just a hydrogen, that's your one that you need to compare on the other side, your OH. Is it up or down? So see how the OH is down and this is down, both on the same side, like sisters, like the cis, cis on the same side, B sisters, so this is B fructose. I don't know how much you have to identify this and this, but you would need to identify that this is a glucose from that, not a galactose, and this is a fructose. So if you can do glucose and fructose, then that's probably enough. So what's the formula? So you've got, they're both of them C6H12O6, but when you put them together, when they react, it's a condensation reaction, so you're going to lose a water molecule. So C6, you double it. H12 would be 24, but you lose 2 because you've lost H2O. Double this, C6H12O6, so that would be 12, but you're losing 1 for the water molecule. So when you put them both together, you have to remember to lose a water molecule. Okay, so what do we call this bond? Technically it's an ether bond, so it's got a carbon, oxygen, carbon, but they're accepting the biology word, so we need to remember this. I should highlight this now, not this. So it's a glycosidic bond. Okay, so this is also in your data booklet. This is called lactose. So it's got 2 6-carbon rings. So are they 2 glucoses, 2 galactoses, or 1 of each? So if you have a look at this one, remember we have to look at here. If it's down in the data booklet, the one that's down is glucose. This one's up, so clearly it's not glucose. It must be galactose. Your CH2 and your O, both in this OH, that used to be an OH. So the reaction, when these two join, you've got a hydroxyl group from both sides, and those 2 hydroxyl groups get together and lose a water molecule and end up with just 1 oxygen. So they're both on the same side, same cis, B sisters, so that's a B galactose. If you look at this one, OH was down, so this was a glucose. They're in opposite sides, so this was an alpha glucose. You just need to know, it's a different way of writing it, and it kind of shows you more easily that the OH was up and this OH was down, and that's how you get the bond between them. I should have explained whilst pressing the button. So to work out if they're both up in the same direction, B sisters, so that's B galactose. And the galactose because it's up, not down. And this one, glucose, because that's down. And alpha because they're in the opposite direction. Okay, maybe I'll pause it here and then you can have a look at that. Might make more sense. Alright, the formula of alpha lactose, so same idea. So same as the sucrose. O's, if you see O's on anything, fructose, galactose, glucose, they're all sugars. The O's means sugar. Later on we'll look at enzymes, and the enzymes to break up a lactose is lactase. So if you see an A here, that means it's an enzyme that would break up lactose. Okay. Alright, so maltose, another sugar, it's 2 glucose stuck together, so 2 glucose rather than a galactose and a glucose. Fill in the table that we've been looking at. So we've done the amide bond between peptides, losing a water molecule to form that, and now we've got the glycosidic bond between sugars. This was an OH, this was an OH, and now it's just oxygen linking. Alright, polysaccharides. So all of these 6 carbons are glucose monomers, and to form starch, is it alpha or beta? So as you can see, we've got our CH, we've got on either side of here around the oxygen. That's what you're trying to work out. You've got just CH2, this group pointing up. So let's have a look. Is the OH down or up? It's down. It's in the opposite direction. So it's alpha. So they're all alpha glucose. So you've got two forms of starch, amylose, which is straight, and amylopectin, which is branched. And you can see to make it branched, if you have, instead of the first carbon bonding with the fourth carbon, if you have this sixth carbon bonding, the OH group on it, you can start branching off from it. So amylopectin is more branched than amylose. So they're all alpha glucose, just different bonding. The more branching you have, the more you can get water molecules into these OH groups and make it more soluble. If you have straight chains, then the next straight chain can bond straight on that straight chain and not let water molecules in. So amylose is less soluble than amylopectin. These also form like rings, and the OH is even further trapped inside. So the more branching you have, the more soluble. There in plants, starch is a storage for sugars in plants, but in animals, there need to be more branching, so it's more soluble. And the sugars are stored in your liver and in your muscle cells, where you need to break down the sugar for the energy. Alright, so cellulose. Have a little look at this for a second. What do you notice about the O, where the position of the O is in the ring? It's flipped over here. So cellulose, and have a little look. You've got your O here, you've got your CH2OH pointing up, and your OH is also pointing up in the same direction. Same, like cis, same side, B, sisters, B. So this is a B glucose. So B glucose, when you put them together and they flip over, they make straight change, and the OH sticking up here, this is a longer side, and it will, an OH on the other chain will form a hydrogen bond between them. So these pack in really tightly, forming hydrogen bonds. So you've got this CH2OH, and then the OH on the other chain, forming hydrogen bonds. So they fit really well together. So glucose, B glucose, alternating strands. Sorry, I should pause that so you can write that down. They make nice, well-linked, hydrogen bonded, strong structures. So this is why plants, it's in their cell walls of plants, which is like the bone of a plant, because they don't have bones. So it keeps the plant able to be standing up rather than just a floppy mess. So you can see glycogen's more bond. This one should show branching too. Amylose is not branched and it's insoluble, and amylopectin is branched. How do carbohydrates originally form? Photosynthesis. So basically we use carbohydrates for energy. So we have to break down, we're going to look at breaking down starch when we eat it, breaking it down into its monosaccharides, absorbing it into our blood, and then breaking up the glucose, breaking it up into carbon dioxide and water to get the energy out. So the energy is originally put in, the carbon dioxide and the water are originally put together to make the glucose in plants, and you should know all of this already, and that it's used for energy. And cellulose is used for structure. Alright, so we like to eat these things because they're very sweet, but then if we don't use the energy, we convert them into fat and store them. So it's better sometimes not to eat them. Sometimes I've had issues with this, aspartame. This is in your book, textbook. So we can have it as an alternative to sugar. It's much sweeter, so you don't need as much. So one of these molecules would give you as much energy as a glucose probably, but because they're so sweet, 200 times sweeter, you don't need as many, so therefore you're not putting on as much weight. What do you notice about this molecule? So you've got this CONH, so it's got an amide bond in the middle of it, which is kind of implying that perhaps this could be an amino acid and this could be an amino acid. So if you look them up, you actually find that these are two amino acids. So this is a dipeptide, which is not written in your data booklet, but would be kind of a good question. There's a bit of a problem. An amino acid, this should have a COOH on here, but it doesn't. It's got a methyl group. So if this was a carboxylic acid, now this is an ester, isn't it? So this must have reacted with a methanol, an alcohol, to make this ester. So if you put two amino acids together and a methanol, you get an aspartame. You could look up and find out what those amino acids are. You really should be able to work out the formula. So if you count them around, you should be able to. I'll just pause the video, see if you can work out the formula, and then I'll show you the formula. So remember, adding hydrogens, every carbon has to have 4 bonds. 1, 2, 3, 4. This one's got 2, so it must have 2 hydrogens. Yeah. So work your way around and see on the ring, 2, 3, 4. So there's nothing added to that carbon. This one's got 1, 2, 3. So it should be another one. So see how many. Okay, hopefully you worked that out. How many chiral centers? So these are clearly not, because they've got 2 hydrogens. So you want to have something that has got 4 different bonds. So there's one there. This has only got 2 of the bonds are with oxygen, so that's not. So this is it's symmetrical. So these aren't. So there's only that one and that one. So just 2. Okay, what type of bond is joining the 2 ends of the molecule? What was that called? The 2 ends? What sort of bond is that? An amide bond. So it's a dipeptide and an ester. And you could break it up and work out what the two amino acids were. Alright, can you write a balanced equation for the complete oxidation of glucose? So complete oxidation is another way of saying turning the carbon into carbon dioxide, which has got the highest oxidation number. So remember with our flow chart, as we went right across from alkanes to carboxylic acids, kept going to carbon dioxide, the carbon's being more and more oxidized. So basically, this question is asking for a combustion reaction, isn't it? Turning glucose into carbon dioxide and water. So you should be able to write a combustion or a complete oxidation reaction. So write the balanced equation for the complete oxidation of biodiesel. This is a bit of revision from the fats. And so what is biodiesel? We did this a long time ago. Breaking up your triglyceride with methanol. So it's a methyl ester. Okay, so methyl ester. Making carbon dioxide and water. I haven't balanced that, have I? Okay, so you can't really do it like this. You have to find how many carbons you've got and then balance it. Odd number, so I have to double everything. Okay, so this bit here, when you're working out these, don't forget this oxygen here before you work out those. So there's 2 lots of 2, there's 4 oxygens over here. So when you counted it up here, you would have 102 on the right. Take away the 4 that you're going to use for this, and you will get 98 left. Half of 98 is 49. Okay, can you answer this one? Okay, so 3 glucose, so you're clearly going to have 18 carbons. 3 of these would be 36. But when you think about it, every time you put glucose with another glucose, you lose a water molecule. So, have I got help with that? No. So, 1 glucose, another glucose, another glucose. Between them, you'd lose a water molecule and another water molecule. So, when the 3 get together, you've lost 2 water molecules. So, you're going to have 36 minus 2 lots of 2, 4. So, 36 minus 4 is 32. So, you're looking at that one. And if you worked out the same with the oxygen, 3 6s are 18, minus 2 is 16, so it's C. So don't forget, every time you put them together, you lose water between them. Okay, so this is kind of a trial and error one. Amylase, glucose, all put together. So glycogen is another polysaccharide. Hydrolysis means you're adding the water back, water adding, lysis breaking. So it's the opposite of condensation. So you want your starch, your straight chain starch, broken up into something smaller. So clearly it's not, you may not have remembered what this is, but clearly it's not glycogen because that's another polysaccharide. Clearly it's not cellulose because that's another polysaccharide. And clearly it's not glycerol because that is part of a triglyceride, a fat. So even if you'd forgotten what that was, so that is a disaccharide with 2 glucose, even if you'd forgotten what that was, you could work it out by trial and error.